We are doing our final project on multiple sclerosis. I'll be giving an overview of the disease, and it starts with autoreactive T and B cells that cross the blood-brain barrier. Once inside, they are reactivated by a myelin sheath antigen that is presented on the antigen-presenting cell. This causes a whole immune response to be triggered that attacks the myelin sheath and causes damage. The myelin sheath is insulation for nerve fibers, and this helps transduce signals from the brain throughout the whole rest of the body. But when the myelin sheath is damaged, like in the case of MS, they are replaced by hard patches of sclerotic tissue. And these patches form plaques in the white matter of the central nervous system. So there are multiple patches of these sclerotic tissue throughout the body. The early stages of MS are characterized by episodes of acute inflammation that resolve within a few weeks. These attacks occur every 12 to 18 months. However, over time, these episodes occur less frequently but resolve less completely, and this is known as progression. And it's the consequence of the repeated damage to the myelin sheath. It's characterized by the accumulation of the long-term disabilities. And so this means that MS is not resolved or cleared. There are 3 types of MS. The first type is relapse-remitting MS, which is the most common, and it's characterized by episodes of relapse of the inflammation attacks, followed by remission. And remission is either complete recovery from symptoms, or in some cases the symptoms become permanent. And the 2 other types are secondary progressive MS and primary progressive MS. And the main difference between these 2 and between relapse-remitting is that these 2 do not have the remission stages and they continue to progress. The symptoms of MS vary from person to person as well as in duration and severity, but some common ones include vision loss, unusual fatigue, numb limbs, and memory loss and tension problems, as well as even partial or complete paralysis. Prognosis. The life expectancy is only shortened by a few months, and MS is not fatal. But there is a concern about the quality of life because of the accumulation of the disabilities. 20% of patients with MS will be bedridden, 20% will require a wheelchair. But 60% of all people with MS can walk without assistance. And 1/3 of all patients will have symptoms for only a short while and without persistent disability. Treatments. There are 2 types. The first one is acute therapy, which involves corticosteroids. The effects are that it seals the blood-brain barrier, so those autoreactive T and B cells can't cross that barrier. They improve nerve signal transduction, and they lower inflammation. The outcomes of these effects are that it shortens the symptoms of MS as well as enhances the overall well-being. The second type of treatment is disease-modifying therapy, and this has the goal of reducing the number of attacks in relapse-remitting MS. And here you can see a picture of all the drugs for this type of therapy.
How does multiple sclerosis start? MS starts when the myelin sheaths protecting neuron cells are lost due to an autoimmune response against the patient's own myelin in the central nervous system, or CNS. The exact cause of the disease is not well understood, but current research suggests that MS is a multifactorial disease involving the interaction between a wide host of genetic and environmental factors. Over 200 genetic loci have been identified as associated with MS. The most strongly associated genetic risk factor is the gene HLA-DRB1, which codes for the beta chain of the human MHC class II. In order for MHC2 to bind and present self-antigens such as myelin proteins to activate helper T cells and immune response, the individual must carry the alleles encoding myelin binding capable MHC2. Therefore, some HLA-DRB1 polymorphisms can be a major risk factor for MS. Polymorphisms in the IL-7R gene is also associated with MS. IL-7R codes for the alpha chain of the interleukin-7 receptor found on B cells, T cells, and the precursor lymphoid cells. When bound to IL-7 cytokines produced in the lymphoid organs, The IL-7R signaling pathway promotes the survival and proliferation of B cells and T cells throughout their development. IL-7 also drives differentiation of helper T cells into the subset Th1 cells that activate macrophages and amplify acute inflammatory responses. The single nucleotide polymorphism associated with MS disrupts an expression silencer, resulting in increased expression of IL-7R, thus making the individual more susceptible to acute inflammation. For lymphocytes to travel into the CNS, they must first cross the tightly controlled blood-brain barrier mediated by CAMs, or cell adhesion molecules. The interactions among CAMs, ICAM1, the integrins ITGB2, ITGAM, and ITGA6, as well as CD58, CD2, and CD4 all synergistically confer susceptibility to MS. Integrins are found on the surfaces of T cells, while ICAM1 is found on the endothelium of the blood-brain barrier. ICAM1 binds to integrins to allow T cell transmigration into the tissue. CD58, expressed by APCs, bind to CD2 on T cells to facilitate APC and T cell interaction. Similarly, CD4 on helper T cells act as a co-receptor in T cell activation. CD2, when triggered, also activates expression of the FOXP3 gene, which induces differentiation of helper T cells into the Treg cells that are critical in suppressing inflammation and preventing autoimmune reactions. One environmental factor associated with MS is vitamin D. Vitamin D, when bound to the vitamin D receptor on lymphocytes and APCs, activates the vitamin D response element in the genome. This inhibits expression of HLA-DRB1 and IL-2. This environmental response element facilitates the production of Treg cells and inhibit pro-inflammatory Th1 and Th17 cell production. Smoking is another environmental factor associated with MS. Cigarette smoke can increase acetylation on the histone H3K9. The acetylation on this particular histone location has been found to be associated with immunoreactivity toward oligodendrocytes, who are responsible for the production of myelin sheaths. The acetylation of this particular histone location is also a critical inducer of pro-inflammatory molecules released by M1 microglia, otherwise known as the pro-inflammatory macrophages residing in the CNS. Finally, infection by the Epstein-Barr virus is also strongly associated with the development of MS. The Epstein-Barr membrane protein, LMP1, induces anti-LMP1 IgG antibodies that are cross-reactive with the myelin protein, MBP. Furthermore, the viral capsid protein EBNA2 found on the surface of the Epstein-Barr virus preferentially binds to 44 out of 104 MS-associated gene loci studied, potentially affecting downstream gene expression. Overall, the patient's own immune response exacerbates the autoimmune process in MS. Once leukocytes have been activated against the myelin sheaths in the CNS, memory T and B cells persist in the body to activate adaptive immune response against the self-antigen across the blood-brain barrier. Environmental factors may contribute to the immune system deregulation, causing uncontrolled inflammation in the CNS.
The specific onset of multiple sclerosis is unknown, but there are multiple ways which the disease can progress. The complement cascade can play a role in the progression of MS. The central nervous system is typically considered an immune privileged site, meaning immune cells do not regularly circulate through the central nervous system due to the presence of the blood-brain barrier. This means the central nervous system typically relies on the innate immune system, such as complement, to manage infection and maintain a normal environment. While complement proteins are normally produced by the liver, due to the restrictive nature of the blood-brain barrier, complement proteins are also produced by astrocytes, cells that contribute to the structure and environment of the brain, and microglia, the macrophages of the brain. Oligodendrocytes are susceptible to binding of complement proteins due to a deficiency in complement inhibitor expression. C1q can bind to oligodendrocytes, which can trigger the classical complement pathway. In situations involving brain injury, mannose-binding lectin and phycolins can bind to DAMPs from dying cells to initiate the lectin complement pathway. Both complement pathways converge at the formation of C3 convertase. C3 convertase results in C3b present on cell surfaces. Phagocytic cells then recognize C3b and phagocytose them. C3b can be further cleaved to produce C3d. C3d can then be recognized by B cells. C3 convertase also leads to formation of the MAC, as well as formation of C3a and C5a. C3a and C5a are chemoattractants that recruit phagocytic cells such as microglia to the site of damage. C3a and C5a also promote inflammation and cause macrophages to produce reactive oxygen species. The inflammatory environment causes damage to other central nervous system cells, causing the production of more DAMPs and continuing the cycle of inflammation. The inflammatory environment causes damage to the blood-brain barrier, and it can become leaky. This allows for more complement proteins, autoreactive T cells, and autoreactive B cells to enter the central nervous system. It has been shown that CD8 positive T cells are the predominant T cell type in MS lesions. MHC1 is highly expressed on inflammatory cells, neurons, and glia in MS lesions. The autoreactive T cells attack the myelin sheaths and continue promoting the inflammatory environment present in the brain. Autoreactive B cells can produce antibodies against myelin sheaths, which allow for recognition by phagocytic cells or can initiate the classical complement pathway. There has been some evidence suggesting that antibodies against Epstein-Barr virus, which causes infectious mononucleosis, display a high antigenetic similarity to myelin proteins and can contribute to the progression of the disease. It's also been found that inflammation persists in the central nervous system despite not having a leaky blood-brain barrier in progressive stages of the disease, leading to the belief that the blood-brain barrier can be repaired and inflammation can continue behind a closed-off central nervous system. This can occur due to the formation of lymphatic-like tissue compartments in the brain that resemble B-cell follicles. B cells, plasma cells, and dendritic cells have been found in these dense B-cell follicle clusters and are widely present in patients with severe MS. Overall, the damage to myelin sheaths is what causes progression of the disease.
Hi everyone, so in my part I will be explaining the impacts and discussing further research on MS. So, as for the social impacts of MS, MS results in a decline of physical and cognitive function, which has an effect on all aspects of a person's life. Not only that, but MS can be perceived as stigmatizing and isolating, resulting in a decline in emotional health. So, who benefits from this information? Well, organizations such as online community websites benefit because they're seeking to create a safe space for people with the disease or where people with the disease can come together and feel connected. Additionally, organizations that collect and analyze data on diseases also benefit. So, that's social. And then economic impacts. So, treating MS is without a doubt very expensive. Lifetime costs exceed $4 million, and this is a concern for organizations who are responsible for paying the cost of these therapies. Furthermore, costs are only increasing due to the expansion of the disease modifying therapy market. This is an even bigger concern for those in developing countries who cannot afford this treatment. And so, speaking of developing countries, the global impact is extremely important to understand. However, information on MS worldwide is scarce. One thing for sure is that MS is a growing global problem and there is widespread inequality between high-income and low-income countries. Yes, there is an increase in neurologists, MRI machines, funding in MS organizations. However, this is only in high-income countries, while in developing countries, these numbers are extremely low or even zero. So, who benefits from this knowledge? Well, policymakers, health professionals, and MS organizations can all make use of this data to ensure equality and improvements of treatments regardless of where patients live. And so, let's look at further research and open questions. And so, as for the open questions, the cause of MS is currently unknown. And however, environmental factors could be related and are currently being studied. And so, most environmental factors have already been disproved and are listed right here. And so, the next question would be, what emerging therapies are more effective than the ones currently available on the market? One such therapy would include monoclonal antibodies, which we have discussed in class and have been shown promising in clinical trials. However, there are risks involved, including opportunistic infections and onset of malignancies. And we can utilize ELISA testing to investigate the effectiveness of these antibodies. And finally, lastly, let's look at future potential treatments, and which would be neuroprotection and stem cells. So both of these are highly active areas of research and have the potential to stop MS from getting worse or even possibly reverse the disability altogether. And so, only with further testing can these trials become a reality. Until then, we can only work to improve the quality of life for those affected by this disease, and hopefully in the near future, MS will eventually become a thing of the past. Thank you very much.